importance, right? They contribute to plant trait evolution over long periods of time, where we see, development of physical defenses like spines in this picture, secondary defenses. Plants and vegetation, or herbivores and vegetation, are locked in this long-term co-evolutionary ballet. And then at a larger scale, herbivores, larger spatial scales, herbivores can influence the relative dominance of plants, and ultimately they can shape the distribution of biomes, so where grasslands are, where forests are. Larger herbivores especially have an important effect on nutrient dispersal and nutrient cycling. They move far distances, they have large capacity in their guts, and they actively counteract nutrient entropy in the Earth system. Herbivores can also have interesting effects with fire and by influencing the way that fuel is distributed on the landscape. And large herbivores have a special relationship with us, in that we different human societies have structured their lives, their experiences, their rituals, and their mythologies around large herbivores. And in particular, we have to talk about the American bison, because my work focuses largely on North America. And bison first came to North America during the penultimate glaciation about over 135,000 years ago. They became established, and after the late Pleistocene extinctions, they became North America's dominant herbivore. There was no other herbivore larger than the bison. And they've performed this keystone role where they were providing habitat for other species. They were promoting key ecological processes, like some of the ones that I just mentioned in the last slide. And the demise of bison has been discussed and is well documented, and you can see the recent Ken Burns documentary if you want to learn more about that. But this story is more about the positive side of things, where it's more about bison's success than it is about their demise. But today, bison are absent from most of their historic range. They're limited primarily to protected areas and private herds. But there's a bright future for bison. There are multiple restoration initiatives aimed at bringing bison back, and there's even potential for landscape scale rewilding, where the Chief Mountain herd, which was just introduced this year by the Blackfeet tribe, is now able to roam into Glacier National Park in a way that even the Yellowstone herd can't move beyond its boundaries very easily. So I want to introduce just the overview of my dissertation and the structure of the talk. We're going to go from a large temporal and spatial scale down to a smaller one over time. So we're going to start with the past 20,000 years as bison antiquus transformed basically into the modern bison, and I'll be looking at fossils primarily for that. And this is about how they respond to long-term climate change at the continental scale. Then the next part will be looking at more regional analysis of the same bison fossil data, but I'll be comparing bison to fire as competing consumers, or just as consumers that have different overlapping ranges. And then finally, the smallest, more local scale project is looking at the application of a new exciting application for fecal biomarkers from lake sediment cores to reconstruct past impacts and densities of herbivores in Yellowstone's northern range. So this first chapter, this will only be a brief summary. I've presented on this work before, and if you want to learn more about this work, you can read the big, long review article that we wrote about bison in North America. But this research was motivated by this question of how has climate change influenced the distribution and abundance patterns of bison since the last glacial maximum. This is when ice sheets were at their greatest extent 20,000 years ago to present day. And the inspiration for this project, finds its origins in this GIF when I was just basically locked up in my apartment during the early days of COVID. I couldn't even go into the lab. So I started playing around with distribution data of fossils. And it started with this animation this obviously brought more questions than it answered. So let's step back here for a moment. And, since I'm so interested in reconstructing past herbivores, I'm going to explain a little bit what I mean by that. So when there are a couple of variables to be interested in here, you can think about herbivore identity, which is related to community composition, to what species are dominant, the quantity of herbivores, which you can summarize as relative abundance, absolute population, or density. And then also you can quantify or characterize their ecosystem impacts. What are they doing to the vegetation? How are they impacting the water, etc. And because we cannot observe paleo herbivores directly, we have to rely on what we call in paleoecology proxies. These are stand-ins for the process that we're interested in. So there are a few different options that we have. We have palynological indicators, which are microscopic indicators of herbivores or herbivory. Coprophilous fungal spores, for example. That's a fancy way of saying mushrooms that grow on poop and the spores they produce. So I'm just here evaluating whether they can help us approach certain variables. Identity, no, not really. They grow on dung, but we can't infer whose dung that it was growing on without other information. And there are some questions about how reliable they are for characterizing herbivore abundance, especially at low densities. Fossils, we know those are. We know that we can usually positively identify a species from a fossil. And whether they can tell us about quantity is dependent on the context and scale at which we're analyzing the fossil record. Sedimentary ancient DNA, which is DNA fragments buried in sediments. It could be in lake sediments or a cave, for example. And, every organism on Earth has a unique DNA fingerprint. So you can, if you have enough DNA strands, you can determine whether an animal was present or absent in your context. And that's more of a yes or no presence or absence indicator. There may be an ability to get what is called pseudoabundance by replicating your samples, but it's not a pure quantitative measure. And now molecular biomarkers, which I'll discuss in the third portion of this talk. These are fossil molecules that originate from organic tissues. They tell you something about the process or the organism that they were derived from, and they're durable enough to persist long periods of time. And they can change in quantity if you have, I'll skip that part, I'll talk about it later. So this is our look at continental scale, past 20,000 years, record of bison abundance. So this line is simply showing the number of bison observations in the fossil record that we have good dates for. And then it's also transformed by a taphonomic correction to account for preservation bias, so the destruction of sites over time. So this is trying to characterize relative changes in bison abundance over time. And I'll just point out a few interesting things that stand out to me. So this is what we call in paleoecology the Younger Dryas. This is a time when in many places the climate reverted to cooler, almost glacial conditions. This is also when many other large herbivores were approaching extinction. So I find it really interesting that bison were actually increasing in abundance through at least the initial portion of the Younger Dryas. And they obviously didn't go extinct because they continued through the Holocene. As the Holocene became warmer, became drier, especially in the bison range, bison abundance decreased. But around 4,000 years ago, conditions in the bison distribution became wetter, and bison abundance responded positively to that. And we also took these bison observations in the fossil record and linked them to simulated climate data in a species distribution modeling framework, which is a fancy way of saying statistical models to correlate environmental variables with bison observations. And we did this at 1,000-year time intervals from 20,000 years ago to present. And so these are just some highlights from key geologic intervals. We have, you'll know the blue areas here show higher predicted probabilities of bison presence. So this is a presence or absence indicator primarily. And we see that bison are hanging out in the southwest, especially near the last glacial maximum. They begin to expand throughout the continents, throughout the western half of the continents during the Bolian-Alarod interstadial. And then by the Younger Dryas, when the ice-free corridor opens up and the Beringian and southern bison populations are able to connect, there's a brief window before the boreal forest closes in where the two bison populations actually intermingle. The bison from the south are primarily moving south or north here. So again, this is that time when other herbivores were struggling, but bison were expanding in abundance. And they go from basically Mexico all the way up to the Arctic Sea and the northern slope of Alaska. Now, and then the Holocene is relatively constant climate conditions compared to what we're seeing up here. And when we get to the late Holocene, we approach what is most like what European settlers were seeing when they were moving across the plains and into the Intermountain West. You'll notice that bison are found in the Great Plains and also west of the Rockies here. And another element that I didn't touch on, but I'll just summarize here, is that we found, based on our climate analysis, that bison prefer milder winters. I know that this is this kind of stereotypical image of a bison these days based on lots of Yellowstone documentaries, but if they had any choice in the matter, bison would prefer not to be pushing aside 2 feet of snow with their heads, and they would prefer warmer temperatures. The temperatures of the 3 coldest months, that was our most predictive variable in that analysis. And bison tended to like warmer temperatures. Okay, so part 2. Now that we had this nice data set of bison observations with clean dates, we could take this and start asking more specific questions about what's going on in this region, what's going on in that region. And here in this lab, we especially have this interest in fire ecology. And bison and fire can both be characterized as consumers. Consumers are organisms or a process here that can physically and chemically destroy vegetation, resulting in energy release. So you can think of combustion and respiration as analogous processes where an organic compound plus oxygen with some catalyst results in the release of water, energy, and carbon dioxide. That is true for respiration and combustion. So we can ask, how do consumer dominance and prevalence respond to spatial and temporal moisture gradients in Holocene North America? So, and then the other part of this question is the moisture gradient. Why are moisture gradients important? So ecosystem types, biomes, different processes can vary across moisture gradients. Moisture availability is one variable that controls the activity of life, essentially. So, for example, you have more photosynthesis as conditions get wetter, as long as other things are held constant up to a point. And then the next step here is to ask, how do past and present consumer dominance patterns in North America compare to less altered consumer regimes of sub-Saharan Africa? So as we'll see as we step through this, we'll notice that, and even if you just look outside and think a little bit critically, you'll notice that North America probably doesn't resemble what it did 200, 300, 500, 1,000 years ago. Whereas modern sub-Saharan Africa, they have much more intact ungulate assemblage and also much more intact fire patterns. They have been changed by human activity a little bit, but not to the same degree as North America. So it provides a good model to compare past dynamics in North America against current dynamics somewhere else in the world. And then this is just to help everyone visualize. Africa has dry, open grasslands, and it's typical to think of large herds of wildebeest inhabiting these. And then when you move to wetter mixed savannas, fire tends to become more prominent in those systems. And it seems that there's a similar pattern in at least historic North America, with bison in drier, open grasslands and fire becoming more important as you move into more wooded environments. So some of our colleagues in Africa have already characterized the distribution of fire and herbivores across moisture gradients. So this is a really another good reason to compare against Africa. And it's a good starting point when we're thinking about what was happening in Holocene North America with these processes. So here you'll see two curves across the moisture gradient. This fire curve here is another visualization of a pretty famous pattern that's been observed by fire scientists and paleoecologists. There appears to be an optimum moisture level for high fire activity. In this case, this is a measure of area burned. But they also had made this interesting step of comparing that against herbivores, and they found that herbivores are dominant in Africa in these drier regions. And there's also, you'll note, a portion of rainfall or moisture availability in the middle here, where both herbivores and fire are prominent, and there's potential for codominance of both consumers. But in Africa, more often than not, it's either herbivores or fire dominating. So this led us to predict that biomass consumption was mainly driven by herbivores in open and semi-arid environments, while fire-dominated biomass consumption in closed, mesic, and humid environments of Holocene mid-continent North America. So what do I mean by those last four words? The Holocene refers to the last 12,000 years of the geologic record, and then mid-continent North America, I'll talk about in a second. But this is an overview figure. This is just a simplification of the pattern observed in Africa, and this is what we're using to inform our predictions for North America, along with little visual representations of what you might observe in these different portions of the moisture gradient. So mid-continent North America is here. I'm referring to these two regions plus the rest of the kind of middle of the continents. We see, okay, let me back up a little bit. All of these observations here, these are nice paleoecological sites that have been developed by other researchers, and the records are available online. And we have sites for charcoal, pollen, and moisture distributed across this. And these regions are occupy different portions of the moisture gradient in North America. We have the northern grasslands, which tend to be drier, and grassland forest transition, which is wetter. And there's minimal overlap between these regions, and you can see that all of these paleoecological sites are distributed across this gradient quite well. So this is an opportunity to look at a dry region versus a wetter region and examine fire and herbivore patterns across that gradient. So a little bit more about the paleo data. We developed proxy records for biomass burning based on standardized charcoal influx into lake sediments, moisture availability from lake level sites in the Rockies, from geochemical signals at Kettle Lake, from dust input at Steel Lake in Minnesota, and also some dune activity in the Sandhills. And then as another representation of moisture availability and just vegetation and fuel structure, we characterize woody plant dominance by looking at the ratio of arboreal pollens and non-arboreal pollen in pollen records. So tree pollen versus grass and shrub pollen. Well, I'm sorry, the grass and the shrub and the tree are AP, and the grasses and forbs are NAP. And then the bison abundance data set that I discussed in my last chapter. So quickly, I'll just summarize what we saw temporally with moisture patterns in the region. This is not the most important point. What's more important is to think about just the difference between the two regions, but the overall difference between the regions, not how they change over time compared to each other. So from 9 to 6,000 years ago, conditions were persistently dry. Between 6 and 4, the proxy records indicate that conditions were generally trending wet but variable. Conditions were wet between 4 and 1.5 thousand years ago, and then the most recent portion of these records indicate some drier conditions and severe droughts up to 500 years ago. And I'll just introduce the data that we use to characterize herbivores and fire in modern North America as well. We use these geospatial data sets to look at these relationships today across a moisture gradient. Okay, so this is a summary figure showing a comparison between our two regions. We have the northern grasslands on this column, and then the grassland forest transition, the wetter region, in this column. We have moisture availability change over time with the best representatives of these different regions, but remember again, these are just to compare within the column. These are not for comparison here across regions. This doesn't mean that this port region is wetter than this region. This is just indicating the trend within the region. And then we also, so we can see that the northern grasslands had lower woody plant dominance. Throughout the Holocene, the northern grasslands were more open, while the grassland forest transition had pretty dynamic change with woody cover, but it was consistently higher than woody plant cover in the northern grasslands. So this is a signal. This is indicating that one region is consistently drier than the other. And that allows us to generalize about the patterns we're seeing here. Over the entire Holocene, we're seeing that bison abundance in the northern grasslands is typically greater than bison abundance in the wetter grassland forest transition. And the opposite is true for fire, with more fire activity, more biomass burning in the grassland forest transition. So if we extract all of these points from the fire record and the bison record, we can see how the regions compare across a moisture gradient, which here is indicated by our woody cover variable. So this is more open, this is more closed, so drier, wetter. And in the northern grasslands, you can see bison abundances are greatest, while fire activity, biomass burning is more typical of the grassland forest transition region. So now that we have these nice curves, we can go back to the Africa data that we based our predictions on and compare. Was mid-continent North America during the Holocene similar to sub-Saharan Africa today? And we do see a similar general pattern with herbivores dominating in the drier regions and fire dominating in the wetter regions, with a crossover at intermediate moisture levels. And this is one reason why we couldn't just directly compare North America in the past to North America today, is because of just how modified North America is today. We have herbivory across this black line, and these are the same scales as the prior Africa plot, where you can see both curves emerge. Here, at this level of analysis, we don't really pick up on any area burned really at all, and herbivory dominates across the entire moisture gradient in North America today. And this is what that looks like spatially. The yellow area of the map. So this, okay, let me back up. This is the proportion of dry matter consumed by either livestock or fire in North America today. And yellow areas indicate near 100% dominance of biomass consumption by livestock. The purple color is indicating near 100% dominance of biomass burning by fire, biomass consumption by fire. So we have a real clear picture of North America, regardless of moisture level, being dominated by livestock herbivory, except for the boreal forests and regions that are mountainous and forested, and rural regions with fire cultures. And this is the modern distribution of, this is that relationship plotted across the moisture gradient, where this is the proportion of dry matter consumed in a given place across a moisture gradient. And there's no point in across the moisture gradient where there's that switch where fire becomes more dominant. It's livestock specifically dominating biomass consumption across the entire moisture gradient. Now that we've established that North America is in some ways similar to sub-Saharan Africa in terms of these patterns, we can ask, or just pose, a kind of a fun, goofy little model, taking the distribution of, biomass consumption by herbivores and fire in Africa, and just putting that into North America based on moisture levels, modern moisture levels. And we see, like what we saw in the paleo data, the drier regions are, based on the Africa predictions, are dominated by herbivores, while the wetter regions are expected to be dominated by fire. There are obviously some limitations to this approach, where, especially further north, there is there's no analog to a tropical savanna, right? This is this is forested, but is relatively dry, which is why it's producing this mixed signal. But it's also so much colder, and the growing season is so much shorter than tropical Africa, that it's really not a good comparison. But I think it is a valid comparison to especially focus on mid-continent North America, where we've established this pattern was present, to look at what things could be or might have been. So in summary, the historical distributions of herbivory and burning in mid-continent North America resemble those of contemporary sub-Saharan Africa, and herbivory dominates biomass consumption in North America today across the moisture gradient, across most of the geography, while fire is now functionally absent from regions where it was once common and important. Okay, part 3. This is the Yellowstone biomarker portion. So 5 years ago, Dave and I were driving to the National Bison Range. I think he knew my rangeland background, so he was like, oh, we have some dry sites at low elevations that are relatively open. Let's put the rangeland guy on those sites. But as we were driving, he was explaining how he was working with molecular biomarkers as a potential indicator for past human presence in the Mission Valley. And, I had this question that popped into my brain. Well, if this works for humans, then why can't it work for herbivores. And it turns out that many other scientists have done work in agricultural contexts and archaeological contexts, looking at domestic species primarily. So there's a nice foundation for the science there. But I wanted to take it to the next level and look at, can fecal biomarkers tell us anything about wild herbivores? And that's the question I asked. Specifically, can we use molecular biomarkers in lake sediments to reconstruct wild herbivore identity and abundance? So our objectives here are to validate the utility of fecal steroids for reconstructing past herbivores, using this in Yellowstone to characterize decadal to millennial scale changes in herbivore abundance and community composition. And then compare those records to multi-proxy paleo environmental data to understand how these herbivores in the northern range shaped and responded to and interacted with their changing environment. This is the moonshot idea, right? Like if all these other things work, then we can get to the multi-proxy comparison and try to bring more context to large herbivores, but there's no guarantee that the first steps would even work out. So I'll introduce the study area here. This is the Yellowstone Northern Range, which includes portions of Montana and Wyoming. It includes portions of the Yellowstone National Park and a mixture of private and public lands outside of the park to the north in Paradise Valley. This is what, if you've ever been to Yellowstone, if you have ever seen images or videos of wild herbivores in Yellowstone, the bison herds, the elk herds, this is what you're most likely watching. This is where many of those species winter, and it's really quite a famous and spectacular place. And my study site in particular is a little lake inside this white boundary. This is the Buffalo Ford catchments, which is near Tower Junction and the lower Lamar Valley. So again, here we have bison, elk, moose, mule deer, pronghorn, bighorn sheep as the main ungulates in the system and the main characters to this story. So allow me just to walk through some Yellowstone history here for a second, with an eye towards ungulate biomass change over time, which is what this plot is showing. We don't have very good records of the populations before, in the case of bison, 1880, or in the case of elk, aerial surveys begin here, and we have some journal records to rely upon through this period. And we can kind of provide our best guess as to what was going on before based on the historical documents and these. So Yellowstone National Park established in 1872, and quickly there were measures to protect these precious herds of herbivores that were being hunted everywhere else. Hunting was prohibited in the national park in 1884. Wolves were actively pursued to make way for livestock production, but also to protect, quote unquote, the animals in Yellowstone. This led to a big population explosion in elk, with populations exceeding 37,000 by 1915, and that led to increased concerns about rangeland degradation and overgrazing. There was a phase of intensive population control and culling of the bison and elk populations from the 1920s to the 1960s. And also through this period, as the populations were being tamped down, there was a transition or a change in thought, a change in philosophy about wildlife management. Kind of a signpost was the Leopold Report, and the National Park Service and Yellowstone National Park began to transition more towards a natural regulation policy where the intensive management of elk and bison ended. We're going to allow these populations to be more naturally regulated. We're going to stop subsidizing them with hay during the winter and trying to control their distribution. And then the final piece came into place with the reintroduction of wolves in '95. So this natural regulation era was accompanied by lots of discussion and thought over, is this the right way to manage the system? Can we even approach a natural system in this way? what about the quality of the range? Are we just going to overgraze and result in big starvation events? Then that led people to ask, okay, what was going on in the past? Can we understand or characterize the herbivore community and their abundance over time before? So this work here builds upon Engstrom et al.'s work, where they cored a bunch of lakes throughout the northern range to try to characterize large herbivore impacts ecologically, but they did not have a direct way to measure local herbivore presence, and also they were limited to about the past 100 years. So this is an extension of that work in some ways, to try to go deeper in time and to more directly measure large herbivores. So yeah, again, this knowledge of characteristics and dynamics of past herbivores is considered critical for a lot of managers, to characterize the historic range of variability, to understand what roles and functions were these herds playing and where were they at what times. But a lot of these questions, people have thrown their hands up and said they'll never be answered. We'll never know. Which is why, well, this is why I'm excited about the science, and in particular these compounds with, these are compounds that everyone produces, all animals produce in their digestive tracts. They're derived from cholesterol, so as they pass through, they're modified by microbes. They're excreted into the environments where they can stay for a long period of time. They can become incorporated into lake sediments, and they can, if we can measure them, they can tell us something about herbivores in the past. So some examples here, we can try to approach measures of quantity. In this example, human population change at Cahokia is represented with a sterol record by this black line here, and it's compared to archaeological independent estimates of population change. So you can see that this is some form of validation where both population changes are reflected in the data. Also, we can approach, or we can begin to characterize herbivore identity. So this study was looking at reindeer camps in Siberia, and they were just taking soil samples and comparing the sterile fecal steroid profiles in the dung to that of the soil sediments to try to characterize who was contributing to the organic matter in the soil. Was it reindeer with winter diets? Was it horses? Was it dogs? Was it humans? They characterized all those species with multivariate analysis of all these compounds. And they found that their soil samples most closely resembled reindeer. So I'm taking this approach to the next level, I think, by looking at not only bulk soil samples, but actually placing the samples in context in time with lake sediments, where we're able to construct models of age across depths in the lake sediment. And that allows us to see change over time. So we poured Buffalo Ford Lake in the northern range. It's grassy with sagebrush, uplands, patches of Douglas fir and pine around in the rocky outcrops, and a riparian edge dominated by lots of sedges, lots of rushes. Here is where it's located again. And so my lab, we specialize in sediment cores, and we can extract lots of information from just the lake mud, including tephra from past volcanic eruptions, pollen, charcoal to characterize fire activity again, and then worked with Dario's lab to characterize biomarkers. Also, I had to characterize what potential sources were contributing to the biomarker signal in the lake sediment. And to do so, I collected dung samples from bison, elk, moose, mule deer, and pronghorn. Now, here's a nice summary of the different compounds that we analyzed and the different ways in which we analyze them. I can't explain it all today. I don't have the time, but I will highlight these two compounds here. These are 5-beta stanols, which are especially indicative of animal sources. Many of these other compounds down here can be produced by other sources, but when we measure 5-beta stanols, we're quite sure, especially when they're in high quantities, that they're being produced by animals. Bile acids also are exclusively produced by animals. So here's a breakdown of the multivariate analysis of these dung samples. I performed a PCA, and then I performed a clustering analysis to see what natural clusters emerge from these distributions. And we found that 3 clusters, one characterized primarily by pronghorn with lots of P's in these samples, a moose cluster, and a cluster characterized, that captured elk and bison together. And the next step was these are the clusters in multivariate space. Again, these are the same. These black dots here are lake sediment samples from Buffalo Ford Lake as supplementary individuals. And this is showing that these lake sediment samples most closely resemble the bison and elk cluster. So the stanol profiles of those lake sediment samples resemble the dung from elk and bison. And that's what that relationship looks like over time. This is the distance of each lake sediment sample to each cluster centroid. And we can see over the past 2,000 plus years, the lake sediment consistently resembled elk/bison dung the most. So that's identity. We've identified that, we've determined that elk and/or bison were the most likely contributors to the lake sediment composition and the steroid profile in it over a long period of time. Now we have quantity, that other variable that we're interested in. And to get at that, we characterize steroid influx. So this is just the past 150 years or so. And the dotted line here represents the long-term average before about 1850. And these are the zoostanols are the 5-beta stanols that I was discussing earlier. We can see that as elk populations were increasing throughout the northern range, the stanol influx was also increasing. The error bars here are the age estimate error bars, so we can see that there is overlap between the peak in elk population and our observation here with a peak in stanol influx. Through the culling phase, levels declined. There was another peak here, which corresponds to relatively low ungulate abundances. But by looking into the historical record, we found that bison were actually wintering in the lower Lamar Valley during this time. So the haying had stopped further upstream at the buffalo ranches, and bison had not yet begun their migrations further down valley. So they were kind of just stuck right around this lake, and we pick up on that signal, I believe, here. And we think that we can identify some herbivore impacts on the surrounding vegetation as well. This willow decline pattern has been documented in other cases, and we see with the explosion, elk populations, willow have declined and that they have not recovered to historical levels. And we also see that phases of high herbivory are associated with less grass and sagebrush pollen production. And this is what some of these relationships look like over the past 2,000 plus years. So this is that dominance record again, just elk and bison dominance relative to other herbivores, where right in your view means they're more dominant and left means less. And we can compare that to the Lamar Cave faunal assemblage, which is based on a fossil record just a few kilometers up the valley, where that study documented presence of elk and bison, so bison being the blue marks here, throughout most of this record, elk show up around 1,000 years ago. And also we see pronghorn, bighorn sheep, and mule deer showing up in the fossil record as well. This is the zoostanal influx plotted on a log scale. So there was consistent input over time until the 20th century, essentially, when that influx level exploded. And I just want to wrap up with some conclusions here. So I think we've shown that molecular biomarkers in lake sediments can be used to identify variability in species dominance, local abundance, and environmental impacts. Elk and bison were continuously present in the Yellowstone Northern Range for the past 2 millennia. Population growth of the ungulates was historically exceptional in the 20th century, and their local impacts were historically exceptional at Buffalo Ford Lake. And I'm really excited, and lots of my colleagues are also really excited about the potential for molecular biomarkers to increase our understanding of long-term herbivore ecology. So just to conclude, I want to say that, just to recap again, when linked with environmental data sets, dated fossils can be used to understand how species responded to changing environmental conditions in the long term, as we saw in part 1, and how species interact with and actively shape ecosystems, as we saw in part 2. And in part 3, I demonstrated how molecular biomarkers can illustrate and illuminate long-term dynamics of large herbivores and other organisms at local scales, and that variability in fecal steroid signatures allows for characterization of dominant species and changes in community composition over time. Now, people tend to use stories about the past to justify their preferred future, and this has certainly been the case with herbivores. We hear that bison were historically absent from this or that region, or that someone's preferred grazing method simulates the way that bison historically grazed. But fortunately, these tools and methods can, as they continue to improve, with continuous improvement, we can better differentiate the truth about these past herbivores from the BS. So thank you again to everybody, family, friends, colleagues, funders, supporting organizations. A special shout out to Julie, my wife, and our little baby boy, Charlie. They're a great inspiration. And I'm happy to take any questions. I saw a hand. I'll repeat it, yeah. Yeah. Yeah, so the question was about part 2 and whether I have any recommendations for prescribed burning. Yes, with acknowledging that these landscapes have been fundamentally changed insofar as people are interested in restoring historic ecosystems and their dynamics, especially in wetter ecosystems. Burning and fire is going to be essential. You can't have tall grass in Illinois without burning.
One more question. We've got a class waiting for question. Online, I think I'll just mention that. Welcome to follow up for anybody. Thanks for coming, everybody. Sorry to left out of here, but we want to make sure.
Thanks.
Grab some food on the way out.